Hey there! Welcome back to Purple Bear Biology. Today we get to talk about one of my favorite topics in chemistry and physiology. We are going to discuss how a cell uses the plasma membrane to control what enters and leaves the intracellular fluid. While this topic sounds deceptively simple, a cell's ability to maintain homeostasis directly depends on the plasma membrane, and for cells like nerve cells, ion transport is an integral part of the function. Today we're going to work on the whiteboard for a little bit. So let's get started by reviewing the relevant information about membrane structure. Recall that the plasma membrane is made up of phospholipids that form two layers. This phosphate bilayer is arranged so that the hydrophilic phosphate heads point out into the intracellular and extracellular fluid where water is. Proteins are embedded into this lipid membrane, and some act as facilitators to transport materials in and/or out of the cell. Water, ions, nutrients, and waste all reside on both sides of the membrane. If a molecule is small and non-polar, then it can squeeze between the phospholipids and make it past the lipid tails. This is referred to as simple diffusion. However, if the molecule is polar like an ion or large like a sugar polymer, then they will have to use a protein to enter or leave the intracellular fluid. This is referred to as facilitated diffusion because it requires a protein to facilitate it. These proteins can act either as channels, like a hallway, or as carriers that actually change shape to move molecules. This is kind of like one of those metal turnstiles you have encountered at a stadium or a subway. Once the molecule they transport binds to the active site, they can change shape and release the molecule on the other side of the membrane. They then revert back to their original shape to perform the task again. The change in shape and movement of the molecules across the membrane often requires energy in the form of ATP to occur. Because of this, facilitated transport can either be passive, requiring no ATP, or active transport requiring ATP. If it helps, you can organize this information into a tree to better understand how transport occurs across the membrane. Overall, we have transport that can be broken down into simple diffusion, requiring no proteins and no energy, and we have membrane-bound transport that can either require no energy, called simple facilitated diffusion, or protein transport that requires energy, called active facilitated transport. Okay, so now that we understand the basics of how transport can occur, let's talk about what influences the direction of molecule movement. Let's draw a membrane just as a straight line, where the top is the extracellular fluid and the bottom is the intracellular fluid. Let's add some oxygen, carbon dioxide, and an ion, like sodium, to the inside and outside of the cell. Notice that the number of molecules on either side of the membrane are not equal. Oxygen and sodium have higher concentrations outside the cell, while carbon dioxide occurs at higher concentrations inside the cell. When we have different concentrations of a solute on either side of a membrane, we refer to this as a concentration gradient. Independently, solutes will always want to move from high concentration to low concentration until they reach equal concentrations on both sides. Small, nonpolar molecules like oxygen and carbon dioxide can pass in between the membrane without a protein or energy utilization, so we refer to this as passive diffusion. If no energy is used, then what causes the substances to move in the first place? The concentration gradient acts as a motivator. You can think of this like a boulder on a hill. The boulder at the top of the hill has plenty of potential energy to roll down if you let it go. Similarly, if you have a concentration gradient and the membrane separating them is permeable, meaning that the solute can move from one side to the other freely, then those molecules will move down their concentration gradient just like a boulder rolling down the hill. Once the concentrations are equal on both sides of the membrane, there will be no net movement of the molecules. But what about the solutes that are polar or large? Well, they still want to move down their concentration gradient, but they will need to utilize transport proteins to change sides of the membrane. Another interesting scenario often exists when the cell actually wants to move the solute against their concentration gradient. This would move solutes from low concentrations to higher concentrations and this requires energy. To understand why, let's visit our boulder and hill example again. Imagine the boulder is at the bottom of the hill. To get it to the top of the hill, you'd have to push it. And pushing a boulder uphill requires energy. Similarly, pushing solutes against their concentration gradient requires energy. So, we call this active facilitated transport. If you are wondering why in the world a cell would do this, you should know that this is one of the primary mechanisms for creating large concentration gradients and then using those gradients to transfer energy as the solute moves back across the membrane. Understanding facilitated transport will be vital for us to discuss nerve cell function later in A&P and cellular metabolism in microbiology. Okay, so you may have noticed that we have only been discussing solutes up to this point, and our amazing water molecules have been waiting patiently to enter the conversation. Water molecules can be thought of as kind of greedy molecules that like to dissolve as much solute as they can. Because of this, they like to travel to the highest concentration of solute they can. When water moves from one side of a membrane to the other, from low concentration of solute to higher concentration, it is a special kind of diffusion called osmosis. While initially this may sound like water is behaving differently than the transport of solutes, really water is just moving down its own concentration gradient. To understand what I mean, let's use an example called a U-tube. On the left side of the tube, we have one sodium atom and 8 water molecules, while on the right side, we have 2 sodium atoms and 8 water molecules. In the middle, we have a membrane that is permeable to water, but sodium atoms are too large to move from one side to the other. Now, initially looking at this, you may think that water will not move because it has the same number of molecules on either side of the membrane. But when we think about osmosis, we have to consider concentrations. We actually have more water molecules per solute molecule on the left side, meaning that water is more concentrated on this side. Because water moves from high concentration to low concentration of water, just like solutes, water would move to the right until it reached equilibrium. Another way to think of this is that the solute sodium has a higher overall concentration on the right side, and water is greedy. It wants to dissolve as much solute as it can, so it will move from the left to the right side of the tube. One important thing to note is that we are dealing with really small values of solute and solvent concentrations, but this is rarely the case in real life. So instead of referring to individual number of molecules in a system, in anatomy we typically discuss a solution solute to solvent concentration in a ratio called osmolarity, also known as osmotic concentration. This value represents the total number of solute particles per unit volume, and is typically listed in units called osmoles per liter. Your cells typically have an osmotic concentration of around 290 milliosmoles per liter. If the solute concentration in the solution increases, then the osmotic concentration increases as well. Solutions with higher osmotic concentrations than our cells are considered hyperosmotic. I tend to remember this because someone that is hyper has too much energy, so a solution that is hyper has too much solute. Conversely, if the osmotic concentration drops to a value lower than 290 milliosmoles, then we would refer to that solution as hypoosmotic. I remember this by recalling that hypothermia is caused by not having enough heat. So hypoosmotic is caused by not having enough solute. When solutions like extracellular fluid have osmotic concentrations equal to the intracellular fluid, then we refer to this as isoosmotic. Hyperosmotic, hypoosmotic, and isoosmotic are all ways to quantify the solute to solvent ratio. However, you may have heard of some similar terms called hypertonic, hypotonic, and isotonic solutions. The latter terms are used to quantify the osmotic pressure by considering the solute molecules only. While the terms do not technically mean the same thing, For the purpose of our conversation today, you can consider them interchangeable when we want to discuss how water will move across the membrane. Okay, so now that we understand solute and water movement across a membrane, let's use an example to discuss the potential physiological impacts of osmotic concentrations in anatomy and physiology. Let's imagine that Fred is a new nurse on staff in your hospital, and he is asked to go replace the IV bags of the patients. He goes to the medical storage cabinet and finds 3 shelves of IV bags. This box represents a sample of each of these solutions. Fred looks at the middle shelf to see if these are the IV bags he was directed to get. Normal saline solution administered in IV bags comes standard at 0.9% sodium chloride. This solution would measure about 290 milliosmoles per liter and be isotonic to red blood cells. He assumes all the bags match the shelf that he checked and loads them all onto the cart. Now let's imagine he puts these solutions into the bloodstream of patients. As I said before, the red blood cell's intracellular fluid measures around 290 milliosmoles per liter. Let's start from the top. The osmotic concentration of 9% saline solution would be much greater than that of the red blood cells. This tells me that the solute concentration is higher on the outside of the cell, making the extracellular fluid hypertonic compared to the intracellular fluid. You may recall that the plasma membrane actually repels ions like sodium, so it would not readily cross into the cell down its concentration gradient. If our solute cannot move, then the water will. But which way will water go? Water will leave the cell because it is greedy and wants to dilute down the salt. What do you think happens when a cell loses massive amounts of water? Well, they deflate like a balloon and die. This would not exactly be doing no harm to our patients, so let's move on to the next solution. Like in the previous example, the red blood cell's osmotic concentration is the same, but now we are administering a solution with a similar solute concentration. Because there is no difference between their osmolarities, water moves out and in at the same rate. This is referred to as no net movement. Last but not least, we have our 0.09% solution. The osmotic concentration in this solution would be substantially lower than 290 milliosmoles per liter, and means that there is less solute dissolved in the solution compared to the cell's intracellular fluid. So, this solution would be considered hypotonic. Which way do you think the water would go in this solution? Again, water is greedy, so it would move to where the solute concentration is highest, and in this case, that would be inside the cell. As water floods into the cell, it will begin to swell. If too much water flows into the cell, it will swell and burst and die. While this example is a little extreme, and you are unlikely to find saline concentrations just laying around the hospital, the situation does help explain how osmotic concentration of the blood plasma could dynamically influence red blood cell function and survival. As a side note, it also shows the importance of a decimal place as well. Simply moving the decimal one place to the left or right can have very dynamic effects on overall concentration and the effects inside of the body. So in this video, we reviewed how the plasma membrane can be used to set up concentration gradients, how movement across that gradient occurs, and lastly, how water is influenced by solutes that cannot readily cross the membrane. I hope you found this helpful. If you have any questions, please drop them in the comment section below. And don't forget to like and subscribe to visit and talk some more. See y'all next time.